Hello, everyone. Welcome to Grand Rounds. Dr. Weissman can't be here today, so it is my pleasure to welcome our speaker. We have Dr. Kenan Onel here. He's a professor of genetics and genomic sciences, the director of the Center for Cancer Prevention and Wellness, and associate director of the Tisch Cancer Institute for Clinical Cancer Genetics and Precision Oncology. He is a physician scientist who studies the genetic contribution to cancer risk and response to therapy. Dr. Onel is known internationally for his work on hereditary cancer and is recognized as a leader in functional genomics, an emerging field integrating genetic and laboratory investigations that is yielding profound new insights into human health and disease. Originally from New York, Dr. Onel obtained a BS and an MA in modern European history from Yale University, and both his MD and PhD in molecular genetics from the Cornell Rockefeller MSKCC Tri-Institutional Program. He completed a pediatrics residency at the Babies and Children's Hospital of New York, Columbia Presbyterian Medical Center, and a fellowship in pediatric hematology oncology at MSKCC. He was then a postdoctoral scholar in cancer genetics at Rockefeller University. From 2004 to 2016, Dr. Onel was a faculty member at the University of Chicago, where he was associate professor of pediatrics, director of the Familial Cancer Clinic, associate director of the MSTP program, and a member of the steering committee for the Committee on Cancer Biology PhD program. He then returned home as chief of medical genetics and human genomics at the Donald and Barbara Zucker School of Medicine at Hofstra/Northwell. Dr. Onel was recruited to the Icahn School of Medicine at Mount Sinai in February 2019. Please welcome Dr. Onel.
Well, thank you very much, Ryan. That was a very kind introduction. And thanks to everyone who's here. It's a real honor and a pleasure for me to have this opportunity to tell you a little bit about clinical cancer genetics. And I hope that you find this of interest. First, I have no conflicts of interest and nothing to disclose. Unfortunately, if any of you would like to change that, please feel free to contact me after this talk. I'm happy to chat. Genetics is an emerging field. With the sequencing of the human genome at the turn of the millennium, there was this expectation that we were entering the dawn of a new era of precision medicine. Excuse me, cancer, of course, is at the forefront of that new era because cancer is the only disease that is defined by having more than one genome. There's the normal or germline genome, which is the instructions for how our cells work. It's exactly the same in every single one of our cells. And then there's the cancer genome that's characterized by acquired or somatic mutations that allow a cancer cell to escape from normal regulatory control. The goal of clinical cancer genetics, the original goal of clinical cancer genetics and genetic testing, has been to really ask the question, why is it that some people and families are predisposed to acquire these somatic mutations while others are not? And, historically, meaning until about 5 years ago, the field was really characterized by being incredibly labor-intensive, time-consuming, incredibly expensive, not reimbursed by insurance, and was largely the purview of counseling. As I hope you'll see over the course of this talk, the landscape of clinical cancer genetics has completely changed in the past 5 years or so. Excuse me. And a lot of that change has really been driven by technology and by cost. Now, Moore's law, some of you may be familiar with. It was an observation by Gordon Moore, who is the founder of Intel, who observed in the field of computers that every 2 years there was a doubling of capacity accompanied by a halving of cost. Now, if we turn to the field of genomics and apply Moore's law, what we would have expected is that with the cost of a single human genome being about $100 million at the turn of the millennium, this is what we would have expected, that now it would cost still well over $100,000 to sequence a genome. But as we can see, the cost per genome and our capacity to acquire the information has vastly outpaced Moore's law to the point where we're now at the era of the $1,000 genome, which has really changed the world. It is, in fact, Moore's law and the Supreme Court that has led to the current landscape for genetic medicine. Um, um, the Supreme Court case is the Association of Molecular Pathology versus Myriad Genetics. Myriad was the first and for many years only genetic testing company that was the field. They felt that by virtue of having all of the information on BRCA1 and BRCA2, they could patent the genes and thereby prevent competition. In 2013, the Supreme Court decided that genes are in fact not patentable. And what this decision did was it opened the door for other companies to enter the field of genetic testing. So thanks to outpacing Moore's law, as the cost for sequencing plummeted, more people were sequenced. As more companies entered the field, the ease of genetic testing increased. It was just business, right? Testing criteria that had historically been designed to maximize specificity, meaning only people most likely to have a positive result were sequenced or had and were analyzed. The criteria were relaxed. Panel testing, meaning looking at multiple genes at the same time, became the norm. And through these innovations, it became possible to perform cancer prevention and surveillance trials. As these results indicated the value of knowing a person's genetic provenance, it became clear to insurance companies that having this information was important in terms of keeping people alive and preventing disease. And so insurance began covering the cost of genetic testing. Companies recognizing that there was an economic windfall in genetic testing worked so that rather than having providers and genetic counselors do all the grunt work to acquire, to do the paperwork and acquire pre-authorization for testing, now it's all handled by the companies and with the goal of making genetic testing as easy as possible. And so thanks to these changes, the landscape of genetic testing has completely changed. This is just one illustrative paper, one of many papers that really points out how the world is different. This is from 2017. It was a paper published by Mary Claire King, who discovered BRCA1 and BRCA2 in 1994 and 1995. And what she did was she and colleagues sequenced 1,000 consecutive Ashkenazi Jewish women with a first diagnosis of breast cancer. She performed panel testing for a number of cancer predisposition genes on these 1,000 women. What she observed was that almost 15% of them had a pathogenic variant or some sort of heritable predisposition to, in this case, breast cancer. What was really striking, however, was that she found that 50% of them had criteria, had family histories that met criteria for genetic testing, but amazingly, 50% did not have family histories. Now, we've always thought of family histories as being our best clue for a person to identify a person who is likely to have a heritable cancer predisposition. In fact, this paper and many others has shown that that's not in fact the case. And as a result of this recognition, there's been a movement towards broader population testing. So for example, now it's recommended that all women diagnosed with breast or ovarian cancer be offered genetic testing at diagnosis. And there's a movement towards testing all Ashkenazi women, irrespective of their family history or their personal cancer status, for 3 founder mutations that are that together are found in over 1% of all Ashkenazi individuals. And so inevitably, the consequence of these insights is that genetic testing has moved from the realm of the genetic specialist to the realm of primary care. It's something that we all know about and something that we all think about. And so the consequence is that clinical cancer genetics is not just genetic testing, but is in fact genetic medicine. The diagnosing of a heritable cancer predisposition has been mainstreamed. Genetic testing has transitioned from having limited clinical implications to having profound medical management consequences, some of which are listed below: cancer surveillance, cancer prevention, cancer therapy, and indeed cancer outcomes are different for individuals with different with heritable cancer predispositions and without. The implications for family planning, and of course, implications for unaffected family members who may be at risk for cancer. And so as the technology for clinical cancer genetics has morphed, it's incredibly important that the field of clinical cancer genetics morph as well from being a diagnostic field to a medical management field. And indeed, pre-test counseling, which has been the norm, must be supplanted by post-test medical management. And knowledge is power. Understanding risk is actually something that patients and their caregivers can take control of and change outcomes. Heritable cancer predisposition is now actually about prevention and wellness. So let's illustrate that with a case presentation, okay? A 33-year-old woman is seen for her annual physical by her primary care doctor. She has no personal history of cancer, but upon history taking, it's observed that her mom was diagnosed with breast cancer at 47. Her maternal grandmother was diagnosed with breast cancer at 60. She had an aunt who was diagnosed with aggressive endometrial cancer at 55. And she has an uncle, same side of the family, diagnosed with pancreatic cancer. Again, she's healthy. She has a healthy sister and a healthy brother and a daughter. So what do we do? First and most important thing to do is to not panic, okay? Because this is actually straightforward, okay? And to sort of help with not panicking, I want to make you all aware of two key resources. First, there was a series of, it turned out that it was put together by an expert panel emerging from pediatric oncology, but a series of 17 papers in clinical cancer research that can be found here that talk about different heritable cancer conditions, their etiologies, and their managements. And then there's a resource on the internet through NCBI called Gene Reviews. This is the link, and essentially what it does is it covers in an up-to-date fashion every single important genetic condition that we care for. So it's a tremendous resource that I use every day and that I really recommend to you as well. But before we start talking about our patient and what to do for her, there's a little bit of education, there's a little bit of language that we all need to understand. And so let's go back to school, Genetics 101. So the genome, which is exactly the same in every single one of our cells, is comprised of DNA, bases, or nucleotides. We're going to use those terms interchangeably. It's the sequence of A, C, T, and G that we all learned about back in 9th grade. Our genome is organized in 46 chromosomes. That is 22 pairs of autosomes, numbered 1 through 22, and 1 pair of sex chromosomes. Females are XX, males are XY. We have 2 copies, as you can see, of every single chromosome and therefore every single gene, one of which we inherit from our mother, the other of which we inherit from our father. The exome, which is the business unit of the genome, is the genes. Okay, we have about 20,000 genes in our genome, but interestingly, the genes themselves comprise only about 1 to 2% of the entire genome. Okay, if we think about the string of A, C, T, and Gs that we were just discussing, it turns out that there are variants that exist in each and every one of us. Where I'm a T, for example, someone else might be an A. And in fact, variation is quite common. Of the 3 billion bases in our genome, each of us has somewhere between 10 and 30 million variants. The reference allele, or the common variant, is referred to as the major allele. The alternate allele, or the one that's found in the minority of individuals, typically is referred to as the minor allele, okay? If we sort of think about how we categorize minor alleles, common variants are found in more than 1% of the population. Rare variants are found in less than 1% of the population. And it turns out, with very few exceptions, variants that are associated with an increased risk of cancer are found in less than 0.025% of the variation. So the point is that there's significant negative selection against cancer predisposing variation in the genome. If we then sort of think about how these different types of variants might be associated with cancer risk, we can think about variants as being very rare to very, very common, and as having a low risk influence on disease risk versus a high influence for disease risk. Now, if we think about common variants, the ones that are found in more than 1% of the population, sort of by definition, because they're common, they can't have a very significant influence on disease, right? Because if they did, then every single person, or the vast majority of people with that variant, would have that specific disorder, okay? In contrast, rare variants can have very significant effects on disease. And when we think about heritable cancer, in fact, what we're looking for are these rare variants that have high effects, that have significant influence on disease. Increasingly, it's becoming clear that there are rare variants, typically found in less than 1% of the population, that do have a significant, though not sort of like an on-off switch, effect on disease risk, okay? And in fact, the bulk of cancer predisposing variation is in that moderate to high penetrance range of variation, okay? So low frequency, moderate effect, low frequency, complete effect, okay? If we think about how geneticists perform genetic studies to identify variants that are associated with an increased risk for disease, there are basically two models. One is looking for common variants that are associated with disease risk. These are called association studies, where we take a lot of individuals with something, a lot of individuals without something, controls, right? We compare either a single variant or across the entire genome to identify variants that are either much more common in cases or much less common in cases as compared to controls. When we identify such variants, they're termed associated with increased risk for disease or associated with protection against a disorder. When that's amortized across the entire genome, then that's a genome-wide association study. The visualization for results of a genome-wide association study are shown here in what's called a Manhattan plot. On the x-axis are the chromosomes numbered 1 through 22. Then on the y-axis is the negative log 10 of the p-value of association. So the higher that negative log 10 of the p-value, the more significantly associated a specific region is with an increased risk for disease. Now, if you think about it, if we're looking for cancer predisposing variation that are found in less than 0.025% of the population, the capacity to do these sort of case control association studies, or these genome-wide association studies, to identify with statistical significance a disease-associated variant is incredibly low. We can't do it. We need essentially endless numbers of cases and controls, which of course we don't have. And so for heritable cancer and other Mendelian disorders predisposing variation, we turn to family studies. This is an example of a pedigree, which is how we visualize families that shows individuals who have BRCA1 pathogenic variations. The way to look at pedigrees are circles are females, squares are males. Vertical connecting lines denote biological siblings. Horizontal connecting lines denote biological parents. Slashes are individuals who have passed away. And the key is presented here at the bottom. And so what you can see is that in this family, the occurrence of a BRCA1 pathogenic variant tracks very neatly with individuals who have the trait of interest, that is, in this case, breast cancer or ovarian cancer. And so we can say here in this family that BRCA1 predisposes to breast and ovarian cancer in this family, okay? Now, again, if we think about modes of inheritance, we all have two copies of every gene, one that we inherit from our mother, one that we inherit from our father. There are two major mechanisms of inheritance: autosomal dominant and autosomal recessive. The autosomal dominant model assumes that one parent is affected and has a pathogenic variant. That parent has a 50-50 chance of passing on to the children either the variant containing gene or the normal or gene or the copy of the gene without the variant. And so what that means is that 2 out of 4 options will 2 out of 4 offspring will be affected just like that parent because of this 50-50 chance, okay? The autosomal recessive model of inheritance assumes that each parent has a variant, but because the variant only manifests when an individual has 2 copies, the parents are unaffected, but they're carriers of pathogenic variants. In this case, as you work out the different combinations, we get a 1 to 2 to 1 ratio, 1 unaffected child to carrier offspring, and then 1 out of 4 kids will be affected, assuming the autosomal recessive model of inheritance, okay? And so these are the 2 major ways that pathogenic variation is passed along from parents to children. Now, if we are in our office and we see a patient, the first question is, who do we test, right? The woman that we saw in our office has a very strong family history, although she herself is unaffected. How do we assess who would benefit from genetic testing? So there are a number of heritable cancer red flags, multiple primary cancers identified in a single individuals, cancer occurring in an individual at an age earlier than is usual, so breast cancer under the age of 50, colon cancer under the age of 50, for example, and then family history, multiple cancer-affected family members, all sharing the same bloodline, and again, a number of whom have cancer. Bilateral cancers, and there's certain pathognomonic cancers that are associated with specific cancer predisposing syndromes that should always raise your antenna as individuals who deserve genetic testing. So Li-Fraumeni syndrome, which is P53, choroid plexus tumors and adrenocortical carcinomas, sarcomas, or low hypodiploid ALL in an individual or their family members, something called Dicer-1 syndrome, pleuropulmonary blastoma, NF1, optic gliomas, and malignant peripheral nerve sheath tumors, NF2, bilateral vestibular schwannomas. If you see any of these in a person or their family members, go straight to genetic testing. If we think about family history, I just want to take a little time, right? This is an example of a sporadic cancer family history. No one in the family has cancer except for a single individual who had colon cancer at a typical age, right, above the age of 50. again, as we saw earlier, this is an example of a family that may raise the question whether there is a variant that is being passed along from generation to generation, right? Here, there is a woman with breast cancer. If we hypothesize that she has a heritable predisposition to breast cancer, then she will have one gene, for example, BRCA1 with a pathogenic variant and one copy of BRCA1 without a pathogenic variant. Each of her children, each of her offspring would have a 50-50 chance of inheriting either the copy with the pathogenic variant or the copy without the pathogenic variant. And we can see she has 5 kids, two of whom had BRCA1 related cancers. And then if we look at the next generation, we see again the occurrence of BRCA1 related cancers in her grandchildren. This individual may have inherited is probably someone who inherited the pathogenic variant, but either has not manifested cancer or has not manifested yet cancer, or is among those who, while having the pathogenic variant, just is fated not to get cancer for one reason or another. That's a phenomenon called incomplete penetrance, where an individual has a risk variant but doesn't have the trait, okay? Now let's take a moment to really dive a little bit more deeply into family history. It is, of course, one of the most important clues for heritable cancer. If there's a positive family history, as we just saw, that's fantastic. However, a negative family history is not so helpful. We saw that in the breast cancer paper that we were looking at earlier. 50% of Ashkenazi women with breast cancer who had a pathogenic variant had no family history. And it turns out that while I just list an example in pediatric cancer that was published in the New England Journal in 2015, this is the case across the board. It turns out that negative family histories just aren't particularly helpful at ruling families out, for cancer predisposition or cancer testing. So why is that? Well, there are a number of reasons. First, families are small. We often don't know things about our own family. They're either just unknown or for various reasons, family members just don't talk about cancer in the families. Family members, of course, can die of causes other than cancer. And so if they die of, say, a heart attack at 55, they're not going to develop cancer at 65, right? Importantly as well, family histories evolve over time. And this is especially the case when the index case or the proband is a pediatric patient. Families are young. And so unaffected could just mean unaffected so far. Finally, this phenomenon that we talked about just a few minutes ago of incomplete or reduced penetrance. And this is something that's becoming increasingly important as we learn more and more about the prevalence of cancer predisposing variation in the general population, as we have the technology and the cost continues to decrease to perform sequencing in vast swaths of the population. What we're seeing is that there are many individuals with cancer predisposing variation who never develop cancer. For BRCA1 and BRCA2, for example, the latest data from a study of well over 60,000 individuals, just was published in the New England Journal in February, showed that the lifetime risk of breast cancer for a BRCA1 pathogenic variant carrier is 50%. Okay, that means by age 85. So that means 4 out of 8 never develop breast cancer, which is really important point to consider. So how do we go about testing? Well, the most important part of performing a test, medical intervention on any patient is informed consent. So education linked with informed consent. And education actually can be performed easily these days through the use of videos, actually, many of which are on YouTube, many of which are quite short but cover all of the key points. Then with education, the informed consent process has a number of criteria. First, a description, purpose of the test, how the test is performed. In the case of genetic testing, it's typically a blood sample, what the results mean, whether there are physical or emotional risks that are associated with the test, whether the results can be used for research, what the results might mean for family members, how and to whom the results will be disclosed, including insurance companies, what happens to the test specimen after the test is complete, the information concerning the Genetic Information Non-Disclosure Act, which is a federal law passed just prior to 2010 that protects individuals against genetic discrimination and protects individuals from being denied health insurance as a result of having a genetic pre-existing condition. And then an acknowledgement that the person to be tested has had the opportunity to discuss the testing with a healthcare professional and have their questions answered, and then finally a signature. So in fact, the informed consent process for genetic testing is quite straightforward. The genetic testing itself is equally simple. We spend, we have spent in the past a lot of time thinking about what the best test is to order for our patients. In fact, it turns out that the major testing labs all have heritable cancer panels, which are gene sets for testing. It turns out that they're very similar, really not much difference one from the next. All of them take on interfacing with insurance, all have a maximum cash pay price with financial assistance programs for patients. It turns out that over 80% of patients actually don't have any out-of-pocket costs. And so, in fact, the bottom line is that genetic testing is now quite easy for our patients, which is fabulous. How does genetic testing work? What does it actually mean to perform a genetic test? Well, as we discussed, germline DNA is the same for all of us in all of our cells. And so the tissue source, which is typically blood or spit, is a biomarker for genomes across all tissues. Our genome is invariant, right, and is the same. DNA is isolated from blood or spit. It's fractured into small pieces, and the regions of interest, meaning the genes to be sequenced, are captured and prepared. The small DNA fragments, the fractured DNA, are sequenced, and each region that's sequenced is covered to high depth, ensuring that the read is accurate, okay? Then these fragments, these reads, are processed, right? They are aligned to a reference genome. Variants, meaning deviations from the reference genome, are identified, and then using a series of informatic tools, pathogenic variants are identified, either based upon American College of Medical Genetics criteria for pathogenic variation, or through or by identifying variants that have been previously identified and confirmed as being pathogenic. It turns out that there are three types of results that will come out of genetic testing. The first is a positive result, that is, a pathogenic variant has been identified in a sequenced gene. A negative result is that a pathogenic variant has not been identified in a sequenced gene. So if you perform genetic testing looking at a panel of, say, 30 genes, and the result comes back as negative, that means that all 30 genes have been actively investigated and no pathogenic variation has been identified. There's also the possibility of an inconclusive result or a variant of uncertain significance. This has historically been the bane of clinical cancer genetics existence. It's a result that's neither a positive or a negative. Maybe it's never been previously observed. Maybe it's been observed in a small number of cancer families, but also in the general population, maybe using bioinformatic tools that predict the pathogenicity of a variant. Maybe they are conflicting, right? Or maybe there are no functional studies for a specific variant to really demonstrate whether it does or does not actually have an effect on gene function. So these are the sort of VUSs, variants of uncertain significance, or inconclusive results. So let's do a little bit of a deep dive, a positive result. So for some positive results, there are guidelines. These are some examples of genes and syndromes that do have guidelines for, now we found this, what do we do, okay? These are really delineated very well in gene reviews that we mentioned earlier, and in the expert panel recommendations, such as the clinical cancer research expert panel series that I mentioned earlier as well. When we perform panel testing, it's important to keep in mind that the results may sometimes be surprising. And so what I mean by that is, if you're testing 30 genes because a patient has a family history of breast cancer, you may find that that patient is predisposed to, say, colon cancer, okay, or thyroid cancer, or something else entirely different. Now, of course, I and most patients would argue that knowledge is power, and it's better to know this than not. But it's important to keep in mind that sometimes there are surprises. Similarly, it turns out that, you know, that in some situations, when sequencing panels are not used to identify pathogenic variation, but rather for other purposes, such as to assess chimerism in bone marrow transplant patients, surprises can occur. I am treating a patient, for example, who is a bone marrow donor for a family member. The family member had chimerism tested, meaning the proportion of the person's, of the bone marrow transplant recipient's genome that was provided by the bone marrow transplant donor, as opposed to being the coming back of the recipient's own bone marrow, which of course is a bad thing, right? And while the patient was found to be 100% donor derived, it turned out that on sequencing, the donor had a pathogenic variant in a cancer predisposition gene that was completely unexpected. So think twice as you're considering using these panels for purposes other than genetic testing, because sometimes things happen, okay? It's important when interpreting results of genetic tests to keep in mind carriers versus affecteds. So reports may list a specific variant as being pathogenic, but if a person, but it may be that they're only pathogenic in individuals who have 2 copies of the autosomal recessive inheritance, right? 2 copies of the pathogenic variant. Heterozygotes may be carriers, okay? So keep in mind that zygosity may play a role in interpreting the consequences of genetic testing results for patients, carriers versus affected individuals, okay? And so what I would argue is that while testing is easy, in fact, surveillance, interpretation, and cascade testing should be performed in consultation with clinical cancer genetics experts. So the field of clinical cancer genetics has really evolved from being focused on pre-test counseling to now being one that is really driven by post-test medical decision-making, okay? A negative result is somewhat easy to understand. it's important to keep in mind that familial is not the same as heritable. It could be that in families, there are multiple cancer-affected individuals, not because of genetic predispositions, but because of shared non-genetic factors. It turns out that especially early childhood exposures and shared childhood environment significantly influence risk for cancer. The other thing is that it's important to keep in mind that cancer is common. 1 in 3 women and 1 in 2 men will develop cancer over the course of their lives. So the co-occurrence of multiple cancer-affected individuals in a single family by chance is not unexpected, given the prevalence of cancer in the population these days. The other thing that's important to keep in mind is that even in individuals or families where there may not be a single on-off switch, a single cancer predisposing variant, such as a pathogenic variant in BRCA1, there are other ways in which genetics can influence cancer risk. We talked about common variants, and remember that individuals who are from a single family share 50% of their variants. So if there is an overabundance in a single family of common variants that are associated with an increased risk for cancer, each individually may not be associated with a significantly increased risk, but in aggregate, having a number of shared variants may in fact together shift an individual's or a family's risk for cancer. So, you know, and I want to really highlight again the importance of chance events, the importance of cancer being common. Even in cancers that are believed to be highly genetic, such as male breast cancer, which is found in only 0.1% of the general population, genetic etiologies are only identified even in these what are believed to be highly genetically loaded cases. Genetic etiologies are only identified in about 20% of male breast cancer cases. And of course, there are other reasons for negative tests, such as technical limitations, testing genes, but not the right parts of genes, and of course, not testing every gene, okay? So it is possible that for individuals who are tested to have a negative result, but still have a sort of undiscovered or unknown cancer predisposition. But having said that, in general, a negative result is actually a positive result, if you will. I want to spend a moment talking about these inconclusive or VUS results, okay? So VUS results are uncertain, inconclusive, right? There's not enough information to definitively classify a variant as pathogenic or benign. What we've learned, however, is that as we follow these variants of uncertain significance and as knowledge is obtained concerning variants of uncertain significance, what we've learned is that over time, over 90% of variants of uncertain significance that can be reclassified are reclassified as benign or likely benign. And this is incredibly important because what it means is that VUS results should not concern providers in most cases. They should not concern patients, again, in most cases, and they should not be used to influence clinical decision-making, okay? Why do we see variants of uncertain significance? Well, we've touched upon that already. We know that heritable cancer risk alleles are by definition very rare. There's a strong negative selection. And so we don't have a lot of information to go on, right? We know that sequencing, so uncovering variants, are biased towards people and families who do have the trait of interest. So there's this selection bias that is inherent in performing studies to identify new variants or new genes that are associated with an increased risk for cancer. And so what that means is that because of this bias, there's limited information on the prevalence of a candidate risk allele in the general population. This is especially the case for individuals and families and risk alleles that are more common in individuals or they're more common in individuals of ancestries that are not Northern European or not Ashkenazi Jewish. And it's just a numbers game. We just don't have enough information about the genomic architecture, the normal genomic architecture, and the occurrence of variation in non-European ancestry populations. So of course, we're going to identify more variants of uncertain significance in individuals of non-European or non-Ashkenazi ancestry. And then, of course, even when we do family studies, first-degree relatives, parents, children, siblings share 50% of their variation just by chance. And so if we're tracking variants within families, many of them are actually going to be tracked within families and found in individuals just by chance allele sharing within families. So the point is, variants of uncertain significance are the bane of clinical cancer genetics, but they really should not be used to influence clinical decision-making. And most of them, when they are reclassified as benign or likely benign. Okay, so now let's get back to our patient. So we perform genetic testing on her and we identify a BRCA1 pathogenic variant. We also identify an MSH6 variant of uncertain significance. MSH6 is a gene that's associated with an increased risk for colorectal and some other cancers. It's a Lynch syndrome gene, okay? And she has this variant of uncertain significance. So now, having performed testing, what do we do? Okay, so based upon the occurrence of a BRCA1 pathogenic variant, we begin mammograms on her. We alternate mammograms with breast MRIs every 6 months. We refer her to a breast surgeon to consider risk-reducing prophylactic bilateral mastectomy with reconstruction. The best results turn out to be in individuals of younger age, between 25 and 30. That's both in terms of improving the success of the surgeries, decreasing the morbidity and adverse consequences of the surgery, and also in terms of cancer risk reduction. We refer the patient to Genome for clinical exams with a transvaginal ultrasounds and CA-125 blood test every 6 months to a year. We offer her a risk-reducing hysterectomy and bilateral salpingo-oophorectomy after about age 40 to 45 when she's done having children. We refer her to GI. She has, remember, a family member, an uncle who had pancreatic cancer, for annual pancreatic cancer screening after age 50 with MRCP and endoscopic ultrasound. Because there is emerging data that there's an increased risk of colorectal cancer in BRCA1 pathogenic variant holders, we recommend colonoscopy every 5 years, starting after age 45. Because there's an increased risk for skin melanoma, cutaneous melanoma, we refer her to dermatology for skin cancer surveillance. Because melanoma can occur in the eyes, ocular or uveal melanoma, we also refer her to ophthalmology for ocular melanoma screening. If she's interested in having other children, we engage our colleagues in reproductive medicine to consider pre-implantation genetic diagnosis linked to in vitro fertilization so that the next generation can be born without the pathogenic BRCA1 variant. Reproductive counseling. Remember, it turns out that BRCA1 is not only associated with cancer risk in and of itself, but in individuals who have two pathogenic BRCA1 variants, they actually have a pediatric onset cancer predisposition syndrome called Fanconi anemia. And 1 out of 4 kids of 2 carriers will have Fanconi anemia. And then we embark upon cascade testing. Each SIB has of our patient, her sister and her brother has a 50-50 chance of inheriting the pathogenic variant. Males are at increased risk for prostate cancer and for male breast cancer, 1 to 6%, as compared to 0.1% in the general population. Male carriers can pass along pathogenic variants to their descendants, 50%, right? And of course, there are other families as well who require genetic testing. As to the MSH6 variant, of course, we don't stress too much about it. So if we sort of step back from this one patient and think more broadly, what is heritable cancer risk in the general population? So again, 1 in 2 men, 1 in 3 women will develop cancer over the course of their life. It turns out that 7 to 10% of all cancer patients have a genetic predisposition to cancer. This is not spread evenly among all cancers, but it seems to be clumped with certain types of cancers. So breast cancer, ovarian cancer, pancreatic cancer, prostate cancer, colon cancer, and endometrial cancer, and then osteosarcomas and pheochromocytomas and paragangliomas are all cancer types that are marked as highly genetic. Now, these are people with cancer. What is the likelihood of their having cancer, a cancer predisposing variant? Now, what about people without cancer? Well, it's amazing. But if you perform questionnaires of women who just go for their annual mammography, it turns out that somewhere between 10 and 20% of all women at the time of screening mammography have a personal family history suggestive of a heritable cancer predisposition. So the implication of this observation is that cancer risk is not just the purview of cancer patients, but is important for general practitioners and has a number of patients that are seen by OBGYN and primary care physicians may be at increased risk for cancer based upon their genetics and just not know it, okay? If we think about here at Mount Sinai, and again, this is now focusing on cancer, right? 10,000 patients are newly diagnosed with cancer who are cared for here at Mount Sinai. Of these, about 6,000 should be offered genetic testing based on guidelines and professional society recommendations. About 10 to 20%, meaning 600 to 1,200 cancer patients per year, will have a positive testing result, meaning an inherited predisposition. Each of these patients, because New York is the world's Roche Motel where people move in, but they don't move out, each patient with a positive result will have, say, 5 first-degree relatives in the New York area, parents, siblings, children. That's another 3,000 to 6,000 patients. Half of these, if we invoke autosomal dominant inheritance, will be genetically predisposed to cancer, 3,000 a year. So what that means is that just at Mount Sinai alone, just based on patients diagnosed annually with cancer, up to 12,000 patients per year require a cancer risk evaluation, and up to 4,200 patients a year annually will require ongoing cancer risk surveillance and medical management. So what that means is that the tests are cheap, easy, and readily available. What we have to figure out is how to incorporate testing into everyday practice, right? How do we scale testing, and how do we ultimately keep our patients safe? And what that really means is how do we manage ongoing surveillance? We talked about our BRCA1 patient. You can see there are a number of different subspecialties that need to be involved and need to communicate in order to keep her safe. How do we systemize cascade testing, meaning testing of at-risk family members? Is there a role in a healthcare system or in the general population for population testing? And importantly, in a system as diverse as Mount Sinai, how do we ensure that there's equity, that everyone who needs testing and deserves testing is offered testing and then has the opportunity to be cared for in an ongoing manner? Right? Equity is a huge issue when we think about all sorts of different cultural, educational, and societal concepts regarding cancer, regarding genetics, and regarding the medical establishment. Okay? So the need clearly is integrated care, system-wide access to testing, a medical safety net for patients who have had testing, ongoing multidisciplinary surveillance, right, across multiple specialties, and then cascade testing and care of family members. And so to fill that gap, to provide patients with that need, we opened here at Mount Sinai the Center for Cancer Prevention and Wellness. And this is just a screenshot from the website that sort of announces us. We actually opened our door just this past August. The idea behind the center is that we serve as a cancer wellness medical home for patients and family at high risk for cancer because of their genetics. The goal is to link testing with ongoing care and to coordinate our patients' multidisciplinary surveillance needs in partnership with our primary care providers and the many specialists at Mount Sinai. Visually, this is what the center looks like. It's this coordinating center, this medical home that patients see as their one-stop shopping for cancer risk. Cancer patients, individuals who come to us through mammography, for example, healthy individuals identified by their primary care physicians who are interested in learning about their cancer risk, family members. And then what we do is we have bi-directional relationships with a variety of hereditary X surveillance and prevention programs that we have put together across the healthcare system. So it's a matrix care model. We are the organizer and the coordinator of care, and we collaborate across the healthcare system with a variety of experts in different domains who understand and care for individuals with genetic predispositions of different cancer types. Importantly, cancer risk is not just about surveillance, it's about wellness. And so we have a variety of experts and social support that help us ensure that our patients stay safe, stay well, and have the resources available that they need. The team is comprised of a number of different groups. The clinical scope is, we think of it as a collaborative programming, serving the healthcare system and patients in partnership. And again, our idea is that we're a safety net for the patients, providing longitudinal medical follow-up for patients with heritable cancer syndromes. We coordinate and navigate the patient's surveillance and help them do what they need to do. Patients have told me time and again, since my time at the University of Chicago, that getting diagnosed with a heritable cancer predisposition was, ironically, the easy part. It's negotiating a complex healthcare system across different specialties that was their problem. And so we aim to help patients with that problem, okay, by reviewing and interpreting the results, facilitating follow-up, facilitating communication, right? And then we complement and synergize with existing wellness programs at Sinai. And we see ourselves as an adjunct to the care that's provided by the primary care provider. And we also see ourselves as the bridge between the primary care provider and the subspecialist who's doing the actual, say, breast or gynonc surveillance. We also oversee cascade testing for family members. And of course, we have a database which facilitates both clinical care and research. This is just a cartoon of the typical patient care. A patient sees the provider, the patient is shown a video and has genetic testing performed. The provider discloses the results to the patient. Typically, we link the results disclosure to a telegenetic consult by a genetic testing company, genetic counselor, that sort of gives the patient a sense of the lay of the land. If the results are negative, we're done. If the results are positive, the patient gets sent to the Center for Ongoing Care when we do that, okay, in collaboration with the healthcare team. And then we work with social work, our family liaison unit, to catalyze testing of family members, which has historically been a pain point for clinical cancer genetics. There are, of course, a number of benefits to the healthcare system that integrates testing and management. It systematizes care, and it allows for the coordination of care throughout the healthcare system by breaking down silos. we get family members, we improve patient satisfactions, it frees specialists to be specialists and primary care providers to be primary care providers. This model, turns out, is scalable across the system because it's reimbursable, and it facilitates the longitudinal follow-up and tracking of outcomes. Tremendous opportunities for research, that goes without saying. And so the key takeaway points from what we've discussed are that clinical cancer genetics is not just genetic testing, it is genetic medicine, care coordination, and providing our patients with a medical home. Thanks to new technologies, comprehensive and inexpensive panel testing can be performed easily, can be done either at point of care or through the CCPW. Because cancer prevention and surveillance involves managing patients across many specialties for life, the centers can serve as a medical liaison between subspecialists and primary care. And of course, we also facilitate testing for family members and their ongoing care as well. And so the goal of the center is to move clinical cancer genetics from this sort of ad hoc mix to a more organized, linear, and, if you will, precise model for risk assessment, disease prevention, and therapy. So who should be referred? Anyone with a positive result, anyone with a positive history, anyone who would benefit from an evaluation. Our goal is to schedule patients within 24 hours and see them as soon as possible. How do you refer patients? There's a central number, can be done through Epic. Here is the center's email. This is my contact information, my email, and my personal cell phone number. We see patients now at Union Square, which is part of the impetus for making sure that you are all aware of the center. We see patients on Wednesdays and Fridays in Suite 4C Union Square. I'm happy to see any of you. If you'd love to stop by, I'd love to meet you and talk to you about the program. We are expanding over the course of the year to Midtown locations, uptown. The goal is to be as close to our patients as possible. And so with that, I will stop. And I don't know if we have time, but if there are any questions, I'm more than happy to try to take them.
Thank you, Dr. Onel. That was very interesting. I think we're at 1:02, so maybe let's just give, if anyone has any very quick questions, go ahead and unmute yourself, or you can put it in the chat. If not, feel free to either email Dr. Onel or myself, and I can connect you with him, and we will end it there. Thank you so much. We appreciate it.
It's my pleasure. Thank you very much. Take care, everyone.
Thank you.